Hello, this is Dr. Paul Cottrell, and I'm recording this on 5/7/2026 at 11:54 a.m. This is a paper that was released in 2014 about the hantavirus glycoproteins. These are the spikes that are coming off of the envelope that assemble to a receptor. We're going to talk about receptors in a different video. This is going to be going into the details of the GN and the GC envelope glycoproteins. Now, let's start with the the hantavirus that's shown on the right here. There is a envelope and a GC and GN glycoproteins that are coming off of the surface, the spikes, and that they are in a tetrameric lobe configuration. So if you're looking down, you'll see it in a four configuration. And like I said in the previous episode, you have these negatively sensed RNA that has three segments, three individual strands, I should say, individual strands. And they are defined as the long, the medium, and the short. And this genome is covered in an N protein. All right, there's an N protein that protects it. And there's also a RNA dependent RNA polymerase that is also packaged with each of the different genomes. Again, a hantavirus has three different genomes, just like influenza has two different genomes. And this is the reason why you can have genetic shift. Now, it's important to understand that to stabilize this strand of the genome, the long, the medium, or the short, you have to coat it with this N protein. Now, the RDRP, the RNA-dependent RNA polymerase, will help to code this into a messenger RNA that is able to produce proteins. And then you have the glycoprotein spikes. Now, the tulip virus is similar. There's a similarity to it. I believe it's called the tulip virus. Let's see. Or the TULV virus has a similar makeup to the hantavirus, except that they have these additional peripheral stalks coming off of the membrane. And you can see that they're underneath the membrane and above the membrane has a different complex compared to the hantavirus. Okay. And with that, we want to go into just a brief overview of the process. Now, again, the virus can dock to the cell surface. There's two versions. It gets endocytosed, gets into a endosome, and there's the early and late endosome phase. And you can see here that there's a pH, and if it's a little bit more gray, then the pH is lower, so it's more acidic. The lighter areas are more closer to neutral, or a little bit slightly alkaline. But the key is that the endosome is supposed to break it apart. Well, the virus has learned through evolution to be able to survive the digestion of this higher pH in the endosome. So this will end up coming out. That what happens in the late endosome phase, the shell or the envelope of the hantavirus is cracked open, and the three genomes, the large, the medium, and the small, end up getting into the cytoplasm. The remaining endosome ends up breaking apart the glycoprotein from the from the virus surface. These are the GC and the GNs. Now there's two paths. One path, it uses its RDRP to turn into messenger RNA. And that messenger RNA will end up docking with the ribosomes that are on the endoplasmic reticulum to produce proteins for the hantavirus. There's another path where what happens is that there's a replication. I didn't want to do that. Didn't want to zoom in there. Okay. There's a replication that takes place, and that replication is getting closer to the Golgi apparatus and assembles in the Golgi apparatus, goes through a series of different stages, and eventually is packaged and exocytosed out of the cell. Now, there's also a method that I talked in the previous episode where the genome that's replicated is chaperoned to the cell surface and then packaged and exocytosed. While the actual glycoproteins for the virus are in an exosome that ends up moving to the cell surface and packaging the viron and then exocytosed. It seems that this method here is more virulent versus the Golgi apparatus method. Okay, now again, there's two types of attachment for the endocytosis. There is the clathrin dependent and the clathrin independent. So sometimes you have clathrin be underneath the cell surface, and that will help with the docking of the hantavirus. And then there's the clathrin independent. Now, I have to research a little bit more, but I believe that one is old world and the other is new world variant of the hantavirus. I will look into that. All right. That's a brief overview of the cell cycle. Now, we're going to go into the details of the actual protein that's created. Now, again, here we have the proteins being created for the GC and the GN proteins that are on the actual surface of the hantavirus. So we're going to actually look at these glycoproteins and how it is created. Now, during this process here, where it's creating the glycoproteins, and starting to move into the Golgi apparatus and starting to get ready for packaging. Right? This protein, this long protein, this GNGC protein is cleaved. And approximately 100, there's approximately 1,138 amino acids. Now, the front part of the protein is what will be the GN, and the back part of the protein will be called GC. And there's cleavage points, and these are cleaved at the SP and the WAASA position to produce two proteins, the GN protein and the GC protein. And as it's being processed in the Golgi apparatus, it's starting to fold properly. All right, there's folding in the there's folding. Let me rephrase this. There's the production of the proteins based on the rough endoplasmic reticulum, and the protein is inside the endoplasmic reticulum and folding. And then it eventually is docked and then released. And that's released, goes to the Golgi apparatus and does further processing to be assembled. Now, remember, this is a tetramer. So there's 4 to each spike. This is a tetrameric lobe. There's 4 of them to make a spike. Now, this is one of the 4. And it gets cut and folded and merged together between the GC and the GN. And then 4 others come in and bind. Now, again, the SP and the WAASA attach areas are the cleavage areas to determine what the GN and the GC are. Now, there's some additional things to keep note. N represents the location of the aspergin resins that are likely to carry glycanization. These are the N's. All right. And at these points, there is glycanization that takes place for the GN and the GC. There seems to be more glycanization on the GN than the GC. Now, in terms of the numbers follow these letters, indicate the corresponding residue member within the GPC of the Andes virus. All right, that's the human-to-human transmission. So these are the areas of concern for glycanization for the Andes virus. Now, the ZF1 and 2 indicates the location of the 2 zinc finger domains, ZF 1 and 2. So this is the zinc finger. And the RNC-BS indicates the suggested ribonucleocapsid binding sites. Which is right here and right here. Now the motif of the YXXL represents residues involved in ubiquitization. And that's right here. Now all this is happening on the GN part of the spike protein. FL indicates the location of the putative fusion loop of the Andes, GC. Molecular structure of the Andes, GN, CT, zinc finger domains, dark cyan. Cyan was obtained from the NCBI databank. And these are the ribonucleocapsid binding is also here at the end of the GN. I'm sorry, the GC protein. All right, what does this all mean? Is that you have processing points that they've figured out to allow for certain binding and certain folding, and the cleavage areas to designate what the GN and the GC are. Now, it's important to remember that the GN and the GC bind together, and they will bind to other GN and GC complexes to build up a tetramer that is this right here. Okay, now, and that's it for right now. That's what I wanted to go over with this video. So this publication in 2014 went over the GN and GC spikes and the glycoprotein areas of the GN and the GC. Please help support the work that I do by making sure you subscribe to all the channels. I have YouTube, Brighteon, Bitchute, and Rumble. If you like the free content that I provide, please share the links. And if you would like to help support monetarily, you can support by being a paid member to my Patreon channel. You can be a member to my Substack channel. You can also donate on my website, the-studio-reykjavik.com. You can donate through Stripe, PayPal, or buy me a coffee and take a look at the great products that I offer. Thank you for listening and have a nice day.